So let's start with our today's lecture. Our today's lecture is particularly based that how a researcher in life science can increase or improve his expertise using certain simple skills and to organize the data, life sciences data. And also I will be teaching, and I will try my best that you should learn that what are the options out there which are suitable perfectly for one particular purpose. And let me tell you one very important thing. So whatever I will be telling you in the next hour or so, these are the products of many hit and trial things. Like I was trying that method, I was trying that portal, I was trying to get information from there and from here and from there. So out of all those many human hours of struggle, I ended up with something very simple and easier. These things looks easier to me, but these are the products of all those efforts which were failed in order to get those things done. So when I taught such things to my student, usually they feel, oh my God, it's so simple. I just need to click there and I will get the answer. I need to just click there and I will get the answer. But remember, I am getting those clicks. I have reached those clicks after the efforts of like a lot of human hours. And over the period of last 10 years, the science, research, internet and information is so robust and I must say is so easily accessible and more versatile that it is like changing at a very faster rate. So all of us, including myself, we need to improve our skills. So the first part of today's lecture will belong to our ability and our method, that how we will going to keep the data of life sciences, how we will going to record and save the DNA sequences and the protein sequences. What are the best possible ways to record the DNA sequences? And of course, we should know from where we can get our DNA sequences. And let me tell you, DNA sequences or nucleotide sequences are of different type out there. You might be looking at the RNA sequence, you might be looking at the gene sequence, you might be looking at the non-coding part of a particular gene, we might be talking about the promoter region, we might be talking about the exons, we might be talking about the introns, we might be talking about the start, stop codons, and a lot of different types of nucleic acids with different formats and different things are out there. So first we should know that from where we can get the information of those sequences more easily. When you keep your sequences, and most of us keep our sequences in Microsoft Word or in the text files. I'm really sorry to tell you that even today, most of us are saving our DNA sequences in the text version, in the text form, or in the Microsoft Word. And I'm really sorry to tell you that this is the most inefficient way of keeping the bioinformatic data or keeping the DNA sequences to work with. You are not supposed to make a record of every gene sequence out there, but you should create records of DNA sequences and protein sequences of the area you are working with. That's all. So that you don't need to go here and there to get those things. You don't need to spend that much time after a month or so, like to get the same sequence once again. You should have a customized folder. You should have a customized DNA sequences with you, which are specifically evolved based on your research. So two things I need to show. The first, how we will going to keep the record of the DNA sequences, and the second, from where we get them. From where we get them, it's like a different options out there, but as a starter, as a starter, I will be using the most famous and common portal, which is called as NCBI, to get the sequence of the nucleotide of a particular gene. I will be sharing my screen from here on with you because I need to switch between the applications. So I am sharing my screen right away. So most of us, we used to keep, first we should know from where we can get the sequence of a nucleotide, and after that I will teach you that how we can keep those sequence of nucleotides, how we can add customize information on the sequence we are generating. Okay, so I will be slow, but the lecture is recorded, so you can always come back to see the websites and to see the tricks which I will be doing in the meanwhile. The first thing, the place I told you, you should go to the NCBI, and I'm not putting any extra keywords so that everybody should go to the most basic platform of NCBI. The NCBI itself is a database, itself is a website which is storing information from all fields of life sciences, all published data, as well as all life sciences information which is generating even today. They are holding and securing and organizing all that data. So we can go and we can get those information more easily. It works like Google. If you understand the Google, I believe you should. Google is not searching using the resources present in our computer. Google is a server placed somewhere else. The server is placed like in the USA. There are lots of computers with trillions of terabytes information about all the web pages. They are present in that server. When we type google.com in our website, it will connect us to that supercomputer, that main server. When we type NCBI, we actually ask that server to browse all the pages that server is containing, for us, and whatever the results we can see, like in the last page, you see all those results. These are the pages which that server has generated for us. So our computer is not doing anything other than that. We are actually sharing the screen with the Google server. The Google server is carrying and containing all that information, not our computer, not the internet. It's their hard drive. So frankly, if the hard drives are not there anymore, if something bad happens to the Google computers, we will not be able to browse all those pages at all. It's not because of the internet, it's because of physical memory they are containing over there. Similarly, when a sequence is published, the NCBI took that sequence and placed it in an organized corner. So when we go to their web portal, we are actually sharing the screen with them. They are sharing the screen with us. We are very thankful for them that we can, get the sequence of any gene. Somebody else work on it. Is the money spent by the, somebody else? We don't believe on them. We don't believe on their religion. We don't believe on anything on them, but we do believe on the sequences they have generated. So we are using their portal, which they have made with all that effort. And you can imagine the amount of effort they are putting in generating these portals. They are spending huge amount of money to keep that portal updated. And when the President Trump came into power, he actually threatened the rest of the scientific community that why we are spending millions of dollars to update the NCBI, and everybody from any country can just go and click and get the information out of it. This is like a thing they are doing for us. They can choose to not to share these things with the rest of the world, but they are sharing. So it's a job. Similarly, the research products or the publications out there, they are the fruits of millions of dollars. And we are getting the results for free. So the people who are like producing those things, they just want us not to have that things freely. So they have created, you know, like the subscription options and other things. So we have to pay and to get that data right away. But I'm very thankful to the people that still the NCBI and the data is like freely accessible. So it can help the rest of the countries to get and for us to get the data out of. The data exists in different categories out there. Different category. Each of the category is like, I mean, amazing information. And I don't know most of them. You are not supposed to know most of them. That is absolutely fine. As a researcher, you need to stick with the tools which are absolutely required for your job. Like if I am an electrician, I need to have a tools which are appropriate for my job. I should be skilled with those tools only. I don't need to have the tools which are related to some other job. Similarly, you need to stick with the particular category which you are using again and again. You should know something about those things. So as I told earlier that I want to get the sequence of a gene from the DNA, the portion, you know, the portion of the DNA which are producing a protein coding sequence, that is called as a gene. I want to get the sequence of that particular portion. So I will choose the gene here, make sure it should be gene here, and then I will type the name of that gene. It's absolutely your choice, whatever the gene you want to try. I will be using a gene, the most famous one, or you can try any. If you want to suggest me, please suggest a gene which I can try. You can text me, okay? I will use example from any one of the, you can just text me for that, okay? Let me know kaun sa gene try kare. I get the first suggestion, insulin gene. Okay, let's type the insulin gene. Anybody knows the symbol of the insulin gene? आप मुझे आंसर कर सकते हैं? If not, I will Google भाई के पास सारे बातों के जवाब हैं. Anybody knows? Before I Google it, I think it is INS. Am I right? I wish my internet, it seems gene symbol is INS. So what I will be doing, I need to search INS. Okay, whatever I'm telling you, these are critical and important thing. It's not everything, but the most important thing you need to remember. So the INS gene is searched out. It says insulin, but insulin is present in the other organisms as well. It is present in the zebrafish. It is present in the cattle. It is present in the pig. It is present in mouse. It is present in bacteria and other species as well. When a gene is written in caps, important, the gene name is written in caps, like this one, INS, most probably they are talking about human genes. They're talking about human gene. When the first letter is in the caps and the remaining in the small letters, that means they are talking about in mouse. But if all are caps, they're talking about in humans. So we should go to the INS in caps, which is insulin and Homo sapiens. This is the location. Somebody asked me the other day, this is a chromosomal location. We will be talking about this thing later on, and some additional information. So I will click on INS. It will take me to the page designed for this particular gene. That page contains a lot of information and this usually boggles our mind. ये हमें kick out कर देती है, यार ये कौन याद करेगा? Remember, you are not supposed to memorize anything. Just be smart and get the things which you need to have out of these things. Since it's the first lecture, so I will be explaining everything like in detail. So it will going to take some time. Okay, please hold your patience with me. So when the information is there, it carries the official symbol and other names as well, and also some short summary of that particular gene. These things sometimes very important for you if you are like working on some XYZ gene, you are working on a particular gene in research, it will let you know ये gene करता क्या है. Very handy little information. You can get that information as well out of it as well. And you can see some related information is there. This one, related information is there. इसको समझना है. This is important. The same gene, like human gene, insulin gene, the information is present in the other portals as well. There exist other portals other than NCBI. There exists a portal called Ensemble. There exists a portal called UCSC browser or UCSC genome browser. There exist other portals which collect and organize the same information. But since the gene is same, so the respectable portals, they usually keeps the record or the code name of the same gene in the other portals as well. So what that means here, Ensemble, ENS, the code start with, after the colon is start with ENSG 0000025 something. This is the code for the same gene on the Ensemble. So that means if you want to study the insulin gene on the Ensemble, this will be the code for the same gene. If you click here, it will take you to the Ensemble page and you can study the same gene in that portal as well. It's a good way so that people do not get confused. So the Ensemble will have a different code on NCBI. So the insulin has a different code on NCBI, but the same insulin has a different code on the Ensemble. And then they have a different code in the other portals as well. So you should keep, if you are working on insulin, you can keep that information or that record for you as well. Then there exists the genomic context, that where that insulin is located on our chromosome. Which chromosome? कौन से वाले chromosome के ऊपर है? Where exactly it is located, from where it is starting, from where it is like ended. The genomic context of that insulin is here. It is like chromosome number 11, NC, and then the code. That code is dedicated for the address of that insulin in human chromosome. You can study the genome browser from NCBI as well. This is the genome browser of NCBI, but I will be teaching you the genome browser from some other place. I will be teaching you the genome browser organized by UCSC genome browser. That is more easy to deal. Okay, so Gavraini, this is like my experience. Based on my experience, I know that getting a particular information is easier on which portal. So the gene information is much easier to get from the NCBI, but genomic information about the genes, locations is much easier from UCSC genome browser. But that does not mean NCBI is not good. It's also good. For some people, it might be more easier. I have a question that the basic information of the gene would remain same. Answer is yes, of course, it will remain same because it's the same gene. But remember, एक portal उसी gene की information को अपने context में अपने style से organize करता है, and the other portal organize the same information in their own way, which they believe is better to understand. So they organize the same thing in a different way. It is like It is like what? It is like different general stores keeping the same products in different ways. एक जनरल स्टोर को लगता है कि this product should be placed here, this product should be placed here, this product should be placed. It will be much easier for the customer to browse. But the other general store believe no, this product should be here, this one should be here. It will be much easier for the, because we are like 7 billion minds out there. So that is why there is a need of multiple different types of organization of the same data. But remember, the codes will be specific to each portal. Then you don't need to worry much about the other things. I'm just highlighting the most important things you should know when you're working on a one gene. Then there is a related articles. Very simple, very easy. If this is your research topic, you can get the related articles here, but that is not all. I mean, you should Google as well, and you should use other portals as well, but you will be able to get the most relevant publications right away. And there are much more. See all 898 citations, which includes insulin. And then they organize the publication in another ways as well. This is an interesting way of organizing the recent publication based on function. Somebody discovered the function of insulin in this way, somebody discovered the function of insulin in this way. So how we came to know about the function of insulin over the period of time, organized in terms of publication. Brilliant organization of data. If this is your research topic, I mean, they are giving you everything. They have already organized the things. हाँ, अगर आप काम कर रहे हैं, if you are working in humans, you need to stick with the publication with the human. You can get an idea from the animal side as well. If you are working in cloning, an expression of insulin in bacteria, I mean, you should pick the publications related to that area, of course, but they have organized these things here for you. Okay, and then I'm taking you down. I'm leaving most of the things which are not required right away. And then there is the GWAS. This is important. I would like to talk on these things as well if times allow us, because we are running short of time. I don't know why, but like as a medical biotechnology, time is running short, running out very swiftly. Okay, so I will taking you down to the most relevant thing which we are discussing right now. That journal gene information. I'm skipping it. I'm skipping, and I want you to go to the NCBI reference sequence. So NCBI reference sequences are the sequences which NCBI has collected over the period of time from different sources, and they considered it as a reference sequence for a particular gene. Here you can find the genomic sequence. Remember, this is the sequence of the genome. Do not confuse it with the genes and other sequence on the DNA present in the chromosome or in the genome. This is the code. Everything starts with NG. Everything starts with NG on the NCBI portal will refer to the sequence of genome. You can get the sequence in GeneBank in faster format. There are different formats. You can get the sequence of the insulin genome out of it. But this is not the agenda. This is not the purpose at this time. Then the next is messenger RNA and protein. This is where I want you to look. Hamara original purpose kya tha? We want to get the sequence of nucleotides which belongs to the gene called insulin. Okay. So here is the first messenger RNA sequence of the insulin gene. The first messenger RNA sequence, 1, and then there is 2, and then there is 3. Oh my God! Then there is 4. Allahu Akbar. So what that mean? That means the insulin gene is present in the form of 4 transcripts. There are 4 messenger RNA which were reported as a messenger RNA of insulin. और आप जानते हैं ऐसा क्यों होता है? You should know. Recall yourself. One gene can be transcribed and then processed in a different way to produce multiple transcripts. Alternate splicing. So these 4 transcripts are the 4 messenger RNA of the insulin gene. अच्छा, अब इनमें से मेरे वाला कौन सा है? I mean, मुझे तो प्रोफेसर ने कहा है कि इंसुलिन पे काम करना है. Oh my God, अब मुझे इन चारों में से कौन सा मैसेंजर आरएनए जो मुझे पिक करना है? That's the most important. और यहाँ पर ब्लंडर करते हैं सारे साइंसदान. I'm sorry, this is a recorded lecture. I need to be a little bit formal with that. Insulin gene, click किया, Google किया, get the sequence, and they have no idea that actually it belongs to the working transcript or a common transcript, or just which transcript they are talking about. The best thing you should know based on your research and publication, that which transcript you are referring for your research. But first, let me tell you the codes. The messenger RNA sequence, I mean nucleic acid sequence, it's not actually the exact messenger, it's a nucleic acid sequence of the gene. One gene is comprised about what? The untranslated region, then the start codon, then the exons, and then the stop codon, and then the untranslated region. So I'm talking about that portion. The code for that nucleic acid sequences is NM. So अगर आपको कहीं पर portal पे NM something code लिखा हो, that means that belongs to the nucleic acid sequence of the gene. If the code starts with NP, it means the protein sequence. So you can get the nucleic acid sequence, DNA sequence, as well as the protein sequence. Now the question remains, चारों में से कौन सा वाला इस्तेमाल करें रिसर्च के लिए? मैंने तो सारा एक्सपेरिमेंट भी कर लिया, प्राइमर भी डिजाइन कर लिए, सब कुछ कर लिया, and then I came to know, oh my God, this is not the functional transcript. I can tell you a jugaad method. The direct method is that you should refer to the publications and see what is the most reported functional transcript. You should browse all of them. But I can tell you, traditionally, it's not the law. Traditionally, the longest transcript variant usually the functional variant of a gene. So I will read the description. Transcript variant. This variant 1 represents the shortest variant. All variants encodes the same protein. So it was reported, but they are like producing the same protein. Okay. This variant differs in the 5' UTR compared to the variant 1. So there is a little difference exists. Somebody reported that sequence as well. But the difference exists in 5' UTR. You should know that thing if you are working as a researcher on that stuff. Number 3, transcript variant. This variant 3 differs in 5' UTR compared to variant 1. All variants encode the same protein. So no problem until today about the protein sequence. So that means the exonic regions are same. Little difference exists in the 5' UTR. So if you are working on the protein product of the gene, you are all fine. But if you are a brilliant researcher and you are trying to study at a 5' UTR sequence, then you need to think that which transcript you are working. लेकिन ये इस लेवल पे होएगा नहीं, हमारे वाले लेवल पे हम तो सिर्फ प्रोटीन बनाने पे, लेकिन but you should think that thing as well. Now the fourth one, transcript variant. This variant 4 differs in 5' UTR compared to the variant 1. All variants encode the same protein. So no problem in all those 4 sequences related to the protein. So I will be using the first variant, the NM. This now note every variant has a different code. NM 000207.3. This one has a different sequence, different code altogether. This will take me to the variant 2 sequence. This will take me to the variant 3. This will take me another code to another variant. So I am using the first variant, this one. Also, you should know certain other things as well. There exists a term called consensus CDS. And also a code is given for that messenger RNA of that nucleic acid sequence. CCDS, consensus CDS. This is another portal. This is another portal, a very big one, but worked under the domain of NCBI. So it is like a branch of NCBI which organize and control the sequences of cDNA. So of course, whatever we will be looking at, when I click here, it will take me to the nucleic acid sequence, that is the messenger RNA sequence. A messenger RNA will be produced out of it, and then that messenger RNA will be converted into cDNA theoretically, or that messenger RNA will be utilized in translation to produce proteins. So whatever the consensus cDNA sequence is there, you know, cDNA sequence is the sequence which is used to create proteins. That will start from where? That will start from the start codon, and that will end at the stop codon. There will be no 5' UTR region. There will be no 3' UTR region. So if I click on the CCDS here, it will take me to a sequence of the same gene. Which gene we are talking about? Insulin. But starting from the start codon, ending in the stop codon, and it constitutes only what? It constitutes only the protein coding regions, which we call as exons. And this is the portion which we usually use in our research, in our thesis, in primer designing, and in all those things. We will go there, but not now. Let's click on the, before the session's time runs out, the NM.
Sir?
G, I have a question, G.
So you said that the longest transcript variant that encodes the functional protein, but in this case, this particular variant, they've mentioned that it represents the shortest variant. So are we supposed to follow it as a rule that the longest transcript variant should be selected, or it's a...
I mean, I told you it's not the law. It's just something which I am telling you. Okay. The best thing is that you need to stick with the publications. They will tell you that which transcript they are talking about. They will give you the sequence and you need to check and compare it. But traditionally, the longest variant is the one which people are working on. Here, the first one is set the shortest, but the other are variations. You know, the below 3, all of them are variations of the first one. So that is why I am using the first one. But that is not the law, it's just for the practice. We need to stick with the publication and the sequences they are using. Okay, when I click on the NM, this code, it will take me to the nucleotide sequence of the insulin. Okay, and I will clicking it right now, and it is taking me to that page which is dedicated for transcript variant 1 messenger RNA region of the nucleic acid messenger RNA sequence of the insulin gene. I will explain this page in the next session. अभी हमने ये सीखना है कि डाटा सेव कैसे करना है. अभी हम सिर्फ एक्सेस कर रहे हैं. Okay, so I hope that this session helped you to have some knowledge about that wonderful portal out there. It looks very simple. Everybody knows about the NCBI, but you should know the same things in a different way or in a better way. Okay, so let's take a break, and when we come after the break, we will talk about that how we can use this page and how we will get the sequence out of that page. You can try these things in the meanwhile as well. I will see you after 5 minutes.